Welcome to Chapter 11, where we are going to be talking about cell division, and this chapter focuses primarily on mitosis and meiosis, which I'm sure you've probably seen in some other science class before, so hopefully at least some of this will be a review. I'm going to have this split into several parts, but I'm going to start out by talking about mitosis, and before we get into that, I'll talk just a little bit about basics of the cell cycle and how the cell cycle is controlled. And then I'm going to actually jump to the end of the chapter, which talks about what happens if you get too much mitosis. Basically, you end up with tumors and potentially cancer. And then I will go back and talk about meiosis, which is a special kind of cell division that's used to produce reproductive cells, sperm and eggs. So what is the point of cell division? And there's really three main roles of cell division that you see here. If you are a unicellular, single-celled organism, cell division is probably your primary means to reproduce, right? That would be asexual reproduction. You're simply cloning yourself through cell division. In multicellular organisms like ourselves, really the two main roles of mitosis are growth and regeneration. So, you started life as a single-celled fertilized egg, and now you consist of trillions of cells, and you got there by mitosis. And even as an adult organism, where you're not producing a ton of new cells all the time, mitosis is still very important to repair damage, right? We can't do anything as dramatic as this lizard here that had its tail lopped off and completely grew a whole new tail, but if you cut your skin, you can repair that damage by mitosis, creating new cells to fill in the gap. Now, before you have a cell division, there's a few things that would have to happen. There has to be some kind of signal to initiate that division, right? We really only want cells to divide when they're told to do so, when it's absolutely necessary. We have to copy all the DNA before we have a division. And, if you think about it, if you're going to create a new cell through mitosis, for example, you want that new cell to have a copy of the genome, right? So we have to copy the genome and make sure it gets correctly segregated into that new cell. And then lastly, we have to have a cytokinesis event, which is actually physically separating the cytoplasm to create those two new cells. So we'll talk about all of these things in the course of this chapter. Now, in prokaryotic organisms and bacteria, they technically don't carry out mitosis. They go through what's called binary fission, but it's essentially the same process. Here you see a bacterium. They just have a single circular chromosome, whereas most eukaryotic cells have lots of linear chromosomes. Like, for example, in each of our cells, right, you have 46 linear chromosomes. So simpler setup here with just the one round chromosome. We have to copy that chromosome and then segregate it to opposite ends of the cell, and then have a cytokinesis event to split it in two. And now we should have two genetically identical bacteria here, right? They each have an exact copy of the genome. They should be twins. So that's binary fission in a nutshell. I already mentioned that. All right, so we're going to focus primarily on eukaryotic cell division here. And as I mentioned, it's a little bit more complicated in eukaryotes just because you have lots of linear chromosomes. So there's more to deal with, especially in terms of the segregation part of cell division. And keep in mind, whether you're talking prokaryotic, eukaryotic cells, you have to copy all the DNA before a division event occurs. And you might see chromosomes represented in two different ways in some of the upcoming chapters, so I just wanted to clarify. If you look at the image on the right here, this is two chromosomes, right? 1, 2. And down here, this is still two chromosomes, it's just this is following DNA replication. So, for example, the chromosome on the left, the yellow one, we've copied it nucleotide for nucleotide, right? We've copied that entire chromosome, but the new copy now is still stuck to the original. So we basically have 2 copies there and 2 copies there, but since they're attached, we just consider this one chromosome still. And we call the 2 copies chromatids. So these would be considered sister chromatids here on the left, 2 sister chromatids on the right. And then these are 2 chromosomes. So don't let that confuse you, right? If you see the chromosomes looking like X's, that just means they've already been replicated, and you have the 2 identical copies attached. So as we're going to see when we go through mitosis, eventually these 2 copies are going to separate from each other, and as soon as they do, then they would actually become independent chromosomes. But for right now, because they're stuck together, just consider this one chromosome, one chromosome. All right, so as I said, we're going to focus on mitosis first, and then we'll come back later and talk about meiosis, which is that special kind of cell division for producing sperm and eggs. But really the goal of mitosis is to create cellular clones, right? We want to create new cells that are genetically identical to the parent cell. And before we get into the stages of mitosis, I just want to remind you that mitosis or meiosis are really just a small piece of the overall cell cycle. So we'll dive into mitosis here in a minute, but if you imagine, let's say, the end of mitosis, cytokinesis here, to be kind of the birth of a new cell, that cell is immediately in what's called the GAP1 or the G1 phase, where it's growing and metabolizing. Then enters what's called the S phase, and then G2, and then it would be back to mitosis. But basically, all the cell cycle that is not mitosis or meiosis, the GAP1, the S, the G2, is all considered interphase. Okay, so interphase is divided into these 3 stages. And as I mentioned, in that G1 gap phase, you have the cell growing, metabolizing. In S phase, S stands for synthesis. This is where that DNA replication process happens. Right? So let me just go back a couple slides here. Right? This, during S phase, is where we would go from these single unreplicated chromosomes to these double replicated chromosomes. And in a later chapter, we'll talk in more detail about that process of DNA replication and how that works. And then you have another sort of growth and metabolism phase, G2, here before you go back into mitosis, where the cell is basically getting itself ready for that division, and then you're back to the actual division process again. And you'll also notice this red line down here. This is what's called the restriction point, which is considered a checkpoint. And you can think of this really as kind of a red light, a traffic light during the cell cycle. So the cell is cruising through GAP1 phase, but once it hits this restriction point, it essentially has to stop and kind of take stock and make sure, hey, is everything going okay? You know, do I really want to keep going through the cell cycle? If not, cells can actually exit the cell cycle at this restriction point and enter what's called G0, which is just kind of a non-dividing phase, essentially just chill for a while. You know, if they're getting the right signals and they really do need to continue through the cell cycle, then they can go past that restriction point and continue on. And so there's been a lot of interest over the years in how cells decide whether they're going to go past these checkpoints, these traffic lights or not. And as I'll show you in a minute, it turns out there's actually a bunch of these traffic lights throughout the cell cycle. And you know, that interest comes from the fact that, as I alluded to earlier, if you lose control of the cell cycle, if this happens unchecked, you end up with tumors and cancer, right? So trying to understand causes of cancer has kind of led us to this understanding of how the cell cycle is controlled. And one simple experiment researchers did early on that kind of illustrated the fact that there are some regulatory molecules that actually help control where the cell is in the cell cycle is they did these cell fusion experiments where they would fuse two cells together that were in different stages of the cell cycle. So the cell on the left is in S phase, a little farther ahead than the cell on the right, which is in G1, right? So these, you have one cell that's up here, one cell that's back here. So we fuse them together, you get this weird kind of hybrid cell that has two nuclei. And what researchers could see when they did these experiments is very quickly, whichever nucleus was farther behind, in this case the one that was in G1 phase, would all of a sudden hurry up and catch up to the other nucleus right after this fusion took place. And this happened over and over and over again. So really the only way to explain that is somehow there have to be some molecules, right, that are actually communicating back and forth, right? Somehow this nucleus, there's some molecule that was present in this cell, right, that can tell this nucleus, hey, we need to be in S phase, so get your stuff together and get with it. So somehow there's these regulatory molecules. And of course, as I mentioned, because of cancer, there was a lot of interest in, well, what are those molecules and how do they work? And we're not going to go too deep into this, but I do want you to be aware those controlling molecules are what are called CDKs, so cyclin-dependent kinases. Kinases are a class of enzyme that like to add phosphate groups to other molecules. That's what kinases do. And so this particular kinase has a partner protein called cyclin, hence the name. This is a cyclin-dependent kinase. So we need these two proteins to come together, the kinase with its partner cyclin, they physically bond to each other, and that kind of activates the kinase activity of this enzyme. And now it will basically take phosphates from ATP and can enzymatically add those to other molecules, right? So just as we talked about back in chapter 8, right, enzyme binds to its substrate here, and then it's going to take that phosphate and add it to the substrate. And by adding that phosphate, we've basically changed the shape and potentially the biological activity of this particular molecule here. And so this cyclin CDK, this phosphorylation activity, is basically what's happening here at the restriction point, as well as some of these other checkpoints throughout the cell cycle. You can think of these checkpoints as those red traffic lights, right? So the phosphorylation by cyclin CDK is turning those red lights green, right? So the cell cruises up to the restriction point, sees the red light, has to stop. But if cyclin CDK is present and active, it will phosphorylate something in the cell that will turn that red light green, and then the cell can go off to the next checkpoint. Stops at the red light, cyclin CDK shows up, phosphorylates something, and so on. So these checkpoints and these cyclin CDK molecules are just giving the cell a lot of control over when and even if it goes through the entire cell cycle or not. Okay, so you might think, well, why wouldn't it want to? Well, maybe the DNA in this cell is damaged, or it doesn't have the correct number of chromosomes or something like that, right? There's some big mutation or something that's happened, right? You wouldn't want that cell to keep dividing and producing copies of itself. You would want it to stop. And so again, I don't want to go too deep into this, but at the restriction point, just to stick with that example, the red light, the traffic light, is really this protein called the RB protein, which stands for retinoblastoma protein. So it is physically preventing cells from going into the S phase of the cell cycle, unless cyclin CDK phosphorylates it. So as soon as the RB protein gets phosphorylated, it becomes inactive, and now the cell can go ahead and go into S phase. And so something like that is happening basically at each one of these checkpoints. And the last point I wanted to make here is you might think, well, okay, we've got these checkpoints, but if cyclin CDK is just always present, right, which is what it looks like here, then the cell is just going to always go past all these checkpoints, right? But that's not the case. What happens is the CDK, the kinase portion, is usually always present throughout the whole cell cycle. Basically, cells are just always making CDK. But the cyclin protein is very transient. It will get produced only if and when it needs to be made. And then it can pair up with CDK for a short period of time, but there's other proteins that will pretty quickly chew up and degrade the cyclin. So it shows up, does its job pretty quickly, and then it goes away. All right, so hopefully you got the basic idea. There are these checkpoints throughout the cell cycle. Cyclin CDKs are these key molecules that determine whether or not a cell is going to go past those checkpoints. Okay. And so, what determines whether or not we produce that cyclin and go past these checkpoints? A lot of times that will come down to, was there a signal telling the cell to divide or not? If there was a signal, then yeah, we want to produce the cyclin CDK and go past those checkpoints. If there's no signal, then that means that cell is not supposed to divide. So what do these signals look like? Well, typically, not always, but typically they're going to be growth factors or growth hormones, right? So one example would be what's called platelet-derived growth factor. Platelets are the cells that initiate clotting in your blood. So imagine you cut your arm, right? The blood clots, platelets accumulate at that clot, and those platelet cells will start producing a protein called platelet-derived growth factor, right? They exocytose it, it diffuses away from the platelet cells, and then there would be receptors on the surface of the nearby skin cells that would bind to that platelet-derived growth factor, and that would trigger the whole signaling cascade that would basically end with the production of cyclin, right? So cyclin gets produced, and then the cell will go past the checkpoint. And in this case, that going past the checkpoint is going to lead to more mitosis, i.e. production of more skin cells. That's going to close or heal the wound in that particular case. All right, so just to remind you, as we already mentioned, right, the DNA gets copied before every cell division, before we go and start talking about mitosis. So we have these double chromosomes, right? Each one of these chromosomes has what's called a centromere in the middle. That's also the point where the two sister chromatids are attached to each other, or most closely attached to each other. And another very important thing that happens before mitosis is what I'm going to call DNA packaging or DNA packing. And this is important because we have all these linear chromosomes that we need to move around, right? We have to segregate the DNA into the two sides of the cell. And that's going to be very hard to do if you're thinking of just like quote unquote naked DNA, right? Just that double helix, these just incredibly long DNA molecules. Whereas it's a lot easier if they're packaged up. So kind of the silly analogy I use is, imagine I plop a plate of spaghetti down in front of you and I ask you to like evenly separate that into, exactly separate it into two different plates. So you've got to like carefully pull out each noodle and, without breaking it, and try to lay it on each of these two plates until you have even piles, it would take forever, right? And it's probably going to be impossible. You're probably going to break some of the noodles, break some of your chromosomes, right? That would be bad. Whereas if I gave you a plate of, pile of raviolis, right? Imagine that's like the packaged up spaghetti, right? It's really easy to separate those out into two different plates, right? So it's the same kind of idea here. It's going to be difficult to separate the unpackaged chromosomes, but much easier if they're packed more tightly. And so this comes back to a few key terms here. One is chromatin, which we've seen before. Chromatin is just DNA associated with proteins. And usually the proteins it's associated with are called histone proteins. So we actually wind the DNA around these histones to help package it. I'll show you what that looks like in a minute. And in the process, we end up with these structures called nucleosomes. It's probably easier if I just show you the image here. Okay, so here's our quote-unquote naked DNA up top, right? The double helix that we all know. And we start this packaging process by taking that double helix and wrapping it around these histone proteins. So you end up with like a little ball of these histones. You wrap the DNA around a couple of times, and then there's another histone that comes on and kind of clamps it into place. Then you get a little bit of naked DNA, and then wrap around another histone, and so on. And so this whole structure, the histones with the DNA wrapped around it, is called a nucleosome. A lot of times you'll see this level of packaging called beads on a string. So it sort of looks like a pearl necklace or something like that. And then we take that and we condense it a little bit more, right? You can see that happening over here on the right side of the image. And then we're going to take this fiber and sort of fold it around some other scaffolding protein, the dark line in the middle here. And then we take that and fold it back, and so on, until you end up with this very densely packed chromosome, right? So this is probably what you think about when you think of mitosis and chromosomes, right? It's those little X's, right? So again, we've got our two copies of the chromosome attached to each other, the two chromatids here, right? But this packaged, it's often called the metaphase chromosome, is going to be just a lot easier to move around than, these very long molecules of naked DNA. So this packaging is essential. And now this brings up the question of, okay, well, if moving these chromosomes around is so important, segregation, how do we do that? That they're not just going to magically like float where they need to go. And the answer is we need to provide some force to push and pull those chromosomes. And you may remember the mitotic spindle is what we're going to use to do that. And that spindle is made of microtubules. Remember that was one of the key types of cytoskeleton that we talked about in chapter 5. And the spindle, those microtubules, will grow from these organelles called centrioles that are found in animal cells. These are basically like little seeds for growing those microtubules, growing the spindle. And the centrioles usually exist in pairs. They're always at like right angles to each other, as you see in the image here. And that pair of centrioles is called a centrosome. Okay? So what you're going to see in mitosis is you'll actually have two of these centrosomes that will move to opposite sides of the cell, kind of growing the spindle as they go, growing those microtubules. And then those microtubules are going to push and pull the chromosomes where they need to go. So it looks something like this, right? So here's one centrosome on the left side, another centrosome on the right side. The green lines are the microtubules. And you can see some of the microtubules attached to the chromosomes and some of them don't. The site where the chromosomes attach, is called the kinetochore. So little green spots here. And again, these microtubules can grow or shrink, so as they do that, they can push or pull the chromosomes around. All right, I will stop there and I will pick this up in the next part.